Hi, it's Mr. Anderson, and welcome to Biology Essentials, video number 38. This is on signal transduction pathways. Signal transduction pathways are very important in the action of cells. They're sometimes misunderstood. So I wanted to start with an analogy. So when Jimi Hendrix plays the guitar, he would vibrate the strings on the guitar. Those would then be transduced. In other words, the pickup in the guitar, the electric pickup in the guitar, has magnets and wires inside it. And what it does is it transduces that message of the wires into an electrical signal. It then goes into an amplifier where so signal transduction pathways in cells do the same thing. It starts with a message, and that message is in the form of a chemical message. And then that's transduced into actions within the cell. And also and so signal transduction pathways all work in the same way. There are a couple of ways that their actions work. Sometimes they'll actually modify a protein or change the shape or the conformation of a protein. But mostly what they'll do is there will be what's called a phosphorylation cascade. In other words, a phosphate group, which remember carries energy, will be passed off from one chemical to another, until it eventually has an action. And protein kinase is important in that. And so the example that I'm going to give you today, a message comes in, the message is picked up by a receptor, and so this line you can say is the cell membrane on the outside of the cell. That message will dock with the receptor, and when it does that, in this case it'll change the shape of the receptor. The example was actually receptor. Then we'll have transduction. Transduction, remember, is when we're switching that message, and we're changing that from a signal message on the outside of the cell to a message within the cell. We use what are called secondary messengers. The one that I'll give you an example of is called cyclic AMP. It's a very common messenger in cells. And then that'll eventually target the cells. In this case, it's going to target cells in the liver, and it's going to make them release glucose from glycogen. And so that's kind of a signal transduction pathway. It starts with a message, and then it eventually has some kind of a target within the cell. This is an animation of this typical liver cell and how epinephrine comes in. So we're going to go through a couple of different spots. So epinephrine is the messenger. Epinephrine is going to be given off the adrenal glands throughout your body, especially affect cells in the liver. So epinephrine is going to dock with this receptor. In this case, it's called a G protein receptor. And so the epinephrine is what's called a ligand. And so a ligand is going to be a chemical. It's a chemical that can't make its way through this cell membrane. Can't make it through this hydrophobic region, so it's going to dock with the G protein on the outside. G protein is a protein that's embedded within. It's actually a sneaky looking kind of a protein. It's embedded within that cell membrane. So it's got a portion on the top, and it's got a portion on the bottom. It's got these units on the bottom. And so what happens is when that ligand attaches, in this case epinephrine, with the G protein, it causes a conformation change in that protein, so it's changing the shape of the protein. Really? What happens when it changes that shape is it actually releases one of those alpha subunits. And so one of the subunits, called alpha subunit, will be released, and it's going to move to a protein just right down the way in the cell. This protein is called adenylyl cyclase. It'll make sense why it's called cyclase in just a second. But essentially what it is before the actual alpha subunit comes, it's an inactivated enzyme. In other words, it's an enzyme that's not working yet. It hasn't changed its shape, so it's actually a functioning enzyme. But once the alpha subunit is in place, it's ready to do its job as an enzyme. In this case, what it does is it converts ATP, adenosine triphosphate, into cyclic AMP, sometimes called this K. Now, what is ATP? When we remember, we know that has three phosphates attached on the outside.
This is an emergency broadcast system.
We're very familiar with how ATP can be converted to ADP when it drops off those phosphates. But it can also drop off two phosphates, and that's what happens.
Is there a disaster? No, it's not disaster.
But there's also a cyclic portion, and so it adds
really.
right where we come off
really.
of
This is so nice of
the
you.
sugar. It's actually going to make a cyclic portion. And I'll put a picture.
You find music like this here. What's going on? This is our family right there.
And now we've created
I'm
these
not
messengers.
down here.
Messengers are going to spread throughout the cell. This is called cyclic AMP. And the secondary messengers, in this case, are going to target something called the protein kinase. Protein kinase, it's made up of a number of different subunits of protein. But kinase means it does something, it does action. In this case, this protein kinase is going to have two catalytic subunits. Catalytic means things that are going to speed or speed up chemical reactions. And then it has these two regulatory subunits. And so as long as the regulatory portions are attached to the catalytic portions, protein kinase is inactivated. Let's watch what happens to the cyclic AMP. It'll actually bind to those regulatory portions of the protein kinase, and it releases the catalytic portions. And so now we have this cascade. In other words, we have this cascade of energy. Those catalytic portions are going to become phosphorylated. In other words, they're going to pick up energy from ATP, and they're going to become activated.
We don't like vacation anymore. It's Jeff.
They then can act on enzymes within the cell. Sometimes they'll act through a number of different cells, molecules. In this case, it's going to drop off that phosphate to phosphorylase, and it's going to activate phosphorylase and release glucose from glycogen. Now, once we don't have that ligand attached anymore, we don't make that cyclic AMP, then the whole thing is going to shut down again.
Let's do it.
The signal transduction pathway is simply a way that we can take this
okay, everyone, let's see now. Join in.
Move it throughout the cell. Desired consequences.
With my hands. Let's join in.
So let's do a little review. And if you've ever watched the show Dora,
are you turning in? Yeah.
There will be times where she just kind of pauses and
I get some kids. I'm hearing kids like this. I get some kids.
So let's do a little.
Hang on a minute.
So we start with
what's going on up there for everyone? Quiet. It's not biggie.
So what do we call this? That's right. It's called the ligand. A ligand is a chemical that can't make entry into the cell.
I'm not really out.
Receptor. So this is called the receptor. Do you remember what that's called? That's right. It's called the G protein. And so what will happen is that ligand will attach to the G protein. It's got a number of different subunits. This one right here on the end is called alpha subunit. Hey, if you're not getting any of these, you may want to go back and watch the earlier portion of the video. We've got this over here. This is probably the one that you're going to struggle with the most. What's the name of that? That's right, adenylyl cyclase. And so what happens is the alpha subunit is going to attach to adenylyl cyclase. We then have an enzyme that's functional. It's going to take in these little starbursts.
You're ready.
That's right, ATP. ATP will then be converted to cyclic AMP or CAMP. And cyclic AMP are now going to work on this.
Don't be nervous.
Protein kinase. That's right, protein kinase. It has two portions that are catalytic and two that are regulatory. That's right. Okay, so the cyclic AMP is going to move over there. Let's watch what happens for a second. Because there's not just a few cyclic AMPs.
Oh, you're number 2 and a number 3. And there's I, the direct injector. The one into the diamonds. I think, don't be shy.
So we just have this one ligand.
Sorry.
We can have all this action. I didn't want to animate all of that, so I went back to just one protein kinase.
Ding dong, ding dong,
free up the catalytic portions.
ding dong.
We now add energy to them. What's that called?
You got this.
Phosphorylation. That's right. We're going to add a phosphate group to them. They're going to turn yellow. And now they're able to pass on that phosphate group.
I'm sure his
phosphorylase. So it's activated. And it can break down glycogen.
There, yay! Thanks so much. You're
So
ready
that's the signal transduction pathway. It's fairly simple.
I'm right
It's got a lot of steps in it.
there.
But it's just like playing the electric guitar.